Okay, so I mean, please, Antoine, would you mind to present our speaker today?
You already mentioned all the admin detail, right?
Yeah, I think it's okay. Just present it in scientifically speaking.
Okay, so it's a great pleasure to have CJ here. Yeah, so I had the pleasure to know CJ many years ago. So he joined our lab back in 2007, 2008, when he was a year 2 undergraduate student in physics at NTU here. So he has spent 3 years in our lab and already have done good research during those years. So very enthusiastic, very passionate about research. And then he did his PhD in NUS, doing single molecule DNA study. And then done 5-year postdoc with Tom Check, Nobel Prize in chemistry in University of Colorado. And he have done a very good work on the structure of protein complex is the telomere. And now he had his own lab in University of Wisconsin-Madison. And just without further ado, please, CJ. Yeah.
Okay. Thank you, David and Antoine, for inviting me to give a very special talk back in my alma mater school in NTU. So today I'm going to talk about our lab research on the human telomere biology. As you can see here, the title is somewhat a mouthful, where we are really using a multi-scale approach to study, how the telomere length is being regulated in the human cells. And you can see here, our lab is now based in the United States, in Wisconsin-Madison. There's a very nice scenery now, but very soon it's going to turn cold and gloomy. But before I proceed to talk about my research, I would like to jump a decade backwards to just because this is a special presentation for me, myself, and for the division. So as Antoine as mentioned, I actually came from, graduated from NTU, SPMS, and this photo is taken in the convocation in 2010. And as you can see that, this is a very small cohort. I think now it's still pretty small for a class in the physics department. And you can see here that this is a very international cohort, which I made a lot of friends from many countries. And I thought one of the highlights during my time in NTU is that I get to meet a Turkish minister together with my friends from Azerbaijan. So you can see here, I actually was the only Singaporean that night in this dinner, and it was really exciting. And I thought the Turkish delight is very sweet for me, but they love that. So as you can see here, the cohort is small, and we gather a lot together to celebrate birthdays like this. So and we still keep in contact a lot nowadays, even with the friends back when we were studying physics together. So a very cool aspect of my undergraduate time in NTU is that we get to do research really early. And as Antoine said, I did my undergraduate research with him for about 2 to 3 years, and I learned a lot of things. And for example, and also had a lot of fun, right? You can see here this, I don't know, Antoine still looked the same to me. That this was actually taken the first, I think, overseas retreat for Antoine's lab. And you can see already there's a lot of group members at the time, and everyone is smiling. And I think that was I still remember like the kind of very engaging scientific discussions that we had when while we are also waiting to go on joyrides in Genting. So this was taken in Genting, Malaysia. Okay, so you sort of can realize that in NTU, at least my time in NTU, I had a lot of fun. I made a lot of lifetime friends, and also it set a lot of bases for my own research philosophies and teaching philosophies down the road. And that's also what I've been will be carrying on in my own lab in Wisconsin-Madison. So now I have to jump back 10 decades in front to current time and back to this talk. So the outline of today's talk is actually just 3 points. First, I will briefly introduce to the audience human telomere biology. Because this is a physics department seminar, I thought I should talk a little bit of the background on this human telomere. And also, and then I have 2 stories that I want to share today. One is on telomerase recruitment, which you will understand what is telomerase and why we're interested in the recruitment, especially in a sense of trying to understand it in a chromatin scale. And actually, the majority of this talk is going to be focusing on this terminator of telomerase, known as the CST complex. And this will be done using a very cool technique that has been a very hot technique recently that emerged in structural biology. So what are telomeres? So telomeres are actually very distinctive DNA protein structures at the end of the linear chromosomes. So because it's only at the linear chromosomes, you're not going to find in viruses or bacteria. But what really draws my lab to study telomeres is because it has a very unique molecular structure. As you can see here, a basic DNA structure, as you guys learned in your high school, is that the DNA are made up of 4 components, A, T, C, and G, and they're made up of 2 complementary strands to preserve the genomic information. And you imagine that most of the chromosomes are made up of double-stranded DNA, but for the telomeres, it's actually made up of 2 kinds of DNA structures, where we have a long double-stranded DNA region that ends with a single-stranded DNA tail, and it ends with the 3' end polarity. But what is also interesting about a telomeric DNA is that both the double-stranded and a single-stranded DNA shares the same DNA sequence. In human or mammals is TTA-GGG. And because this strand is having the TTA-GGG DNA sequence, often people call it the G overhang, which looks like an overhang coming off the double-stranded DNA region. So why did cells or the eukaryotic cells make something so unique or interesting, right? Well, it turns out that the telomeres are very important for our genome stability. As you can see here, this is a very standard metaphase spread of the human chromosomes with telomeres stained pink in colors here. As you can see, every end of the chromosome, you have a pink dot on it. And like I said, telomeres, protects the chromosome ends. And as an analogy, you can think of these like using shoelace, right? That the telomeres are like your anklets at the end of your shoelace, those plastic tips that prevents your shoelace from defraying and damages. But then you wonder, why would our DNA or linear DNA ends get damaged over time? This is because of the end replication problem. So I'll briefly talk about it for those that are not aware of this. So our DNA has to be copied every cell division, right, to preserve the genetic information. So in order to do that, the polymerase, which is the enzymes that use to copy the DNA strand, has to use a primer right here. And then with the primer, it can further extend the DNA here to make the second copy. But then the position of this primer is not guaranteed to fill up right at the end of the DNA strand, the template strand. So that means every time you do a DNA replication, you have a problem of missing a portion of the DNA at the end of the template. That's why the telomere is needed to serve like a disposable buffer during DNA replication. You can see the DNA are now defined as the end of the chromosomes, and as every time the cells divide, you get your telomere shortened over time. So as you can see, when the cells divide, the telomere starts to shorten, and that's why the telomere length is often known as a biological clock for cells. Because if you look at your telomere length when you are a baby, and now the telomere length that you have, you'll find that when you're a baby, you have a longer telomeres than what you have now. And as you grow older, your telomeres will get shorter. So telomeres will get shorter, but what is extending the telomeres? Like I alluded earlier, this telomeric DNA is actually extended by a very special ribonuclear protein known as the telomerase. So the telomere is basically like this title say ribonucleoprotein. It means that it's made up of both an RNA molecule and a protein component. So the telomerase, what it uses is the RNA template to make the repetitive DNA sequence. Over here, you can see they use the complementary strand to make the TTA GGG overhang. So what's so important about the telomere length, right? Because telomere length is heavily regulated in important biological processes. So over here is a very simple chart where the y-axis is the telomere, average telomere length in the cells, and over the x-axis is the number of cell division. As you can see, in the germline cells, which is the sex cells, the eggs or the sperms, the telomerase is activated. And then that's how they can maintain the telomere length over time. But in other cells, non-sex cells, which is somatic cells, the telomerase has to be switched off and the telomeres will be shortened over time because of the DNA replication. Now, of course, when it hits a critical length of the telomeres, when it's short enough, it will hit what is called a senescence or a crisis where the cells will go to programmed cell death. But occasionally, when the cells divide, you will acquire mutations that somehow switches telomerase back on, and that's where you can immortalize cells. And that could be a big problem for biomedical research, which I'm going to talk about later on. But what is really interesting is that these telomeres, because they have both double-stranded and single-stranded DNA, they are able to loop back and form this thing called a T-loop, which actually has the single-stranded DNA invades the double-stranded DNA region as shown here. So it's basically protecting the single-stranded DNA from unwarranted access by telomerase or DNA damage response. You can see here, this is really a simple analogy where the girl is trying to camouflage herself in the background of the white wall from his brother. So telomeric DNA is not going to operate on its own. It's actually being coded by many telomeric proteins. One of these proteins that we are interested in is called the shelterin, which is a very unique protein that's able to bind single and double-stranded telomeric DNA. So shelterin is actually a 6 subunit protein complex made up of TRF1, TRF2, TIN2, RAP1, TPP1, and POP1. But only really the TRF1 and TRF2 binds the double-stranded DNA region, while the POP1 binds the single-stranded DNA. So you can imagine this shelterin complex is perfectly designed to stabilize a T-loop structure such as this, where the double-stranded DNA would stabilize the invaded strand that is now in a double-stranded DNA format, and with the POP1 binding to the displaced strand in red in color here. So far I've talked about a basic introduction to the human telomere biology. Next, I'm going to talk about why we have to understand telomerase recruitment in a chromatin scale. And I'm hopeful that the earlier discussion of telomere biology will make you guys be careful about not trying to go around and extend your telomeres, because in Amazon, we realized that you can actually buy pills to extend your telomeres, or in this case, increase your telomerase activity, which I will say this is not advisable because actually 90% of cancer cells has the telomerase activity in overdrive. You do not want to do this. So this is a very basic overview of the molecular landscape of the shelterin-mediated extension of telomeres by telomerase. As you can see here, you have the double-stranded telomeric DNA, the repetitive sequence bound by multiple copies of the shelterin complex. And then over here, this is where the 3' single-stranded telomeric tail is, and that's where the telomerase is needed to bind and extend the telomeric overhang. So the extension by telomerase are made of 3 major stages. First is the recruitment of telomerase to the telomeres. Second is the actual activity itself, extending the telomeres by telomerase. And then, of course, like I said, we don't want the telomerase to forever extending the telomeres, so it has to be promptly terminated. So for the recruitment, so telomerase is recruited to the telomeric DNA by the shelterin complex. Right, the shelterin complex will bind to the single-stranded and double-stranded DNA region of the telomeric DNA before using the TPP1 subunit to directly recruit telomerase to the telomeres. But we know all these molecular interactions and the players involved in the direct recruitment of telomerase to telomeres. And this is actually done in studies using overly simplified telomeric DNA fragments. But in reality, we have to consider how shelterin directs telomerase to the very 3' end, because the telomeric DNA is long, repetitive DNA sequence. You could have multiple binding configurations of the shelterin complex, which could easily distract the telomerase from finding these 3' single-stranded telomeric overhang. So in our lab now, we're really trying to understand this whole recruitment process in the context of a long telomeric DNA. So our approach is really to use single molecule imaging to study how shelterin is assembled at long telomeres and how these shelterin coated telomeres can recruit telomerase to find a 3' overhang. And these projects spearheaded by one of our graduate students, Josh, in our lab. So for example, one experiment that Josh is trying to do is trying to use atomic force microscope to look at individual DNA protein complexes formed by the shelterin and telomeric DNA. As you can see here, this is a AFM images of telomeric DNA alone. This is the telomeric DNA construct that we use. You can see individual DNA like a wiggly worm. And then now you have TRF2 added into this solution. You can see the TRF2 are now binding to the internal regions of telomeric DNA, because as you remember, TRF2 binds to the double-stranded DNA region, right? And over here, if he includes only the part 1 protein, part 1 binds to the single-stranded DNA. And then you, as one would expect, you see the part 1 now binding to the end of this telomeric DNA construct. So one of our hypotheses is that a fully assembled shelterin complex that both contains TRF2 and part 1 would be able to direct itself to the single-stranded and double-stranded DNA junction. As you can see here, using AFM, single molecule imaging, we can see now most of the shelterin complex is now located at the end of the telomeric DNA. Right here. So Josh also has another goal in our lab. It's really to understand the dynamics involved. What you saw earlier is static images of protein bound to telomeric DNA. But I think the answer lies also in the dynamics involved. How it's being assembled, is the shelterin sliding or hopping along the telomeric DNA, and how this whole process eventually recruits telomerase to the 3' end. So the whole idea is that we'll like to see how this telomeric protein dance along a very long piece of telomeric DNA. So the ideal case, as anyone would like to do, an ideal experiment is that you'll be able to stretch a telomeric DNA and then start to see, your proteins moving along it and study in real time. So to do this, we are going to combine single molecule imaging and manipulation to study how telomeric proteins assemble at long telomeric DNA. And this is done by using a dual beam optical trap in our lab that we can now stretch a telomeric DNA and study how fluorescently labeled telomeric proteins can be assembled. So over at the bottom here is an example of that's using experiment using CRISPR-Cas9 that was done using a similar kind of experiments. As you can see here, is very informative. You can see how individual proteins bind to a DNA that is actually stretched across between these two beads. So I've talked about the second part of my project, so our lab project. So now third, we're going to talk about how telomerase is being terminated by the CST complex. And really, this is a really unexpected discovery that we found when we use cryo-electron microscope to determine the structure of the CST complex. So just jumping a little bit back and resuming to this whole cycle of telomerase extension of telomeres. Like I mentioned earlier, shelterin is needed to recruit telomerase to the telomeric DNA. But shelterin is also able to stimulate telomerase extensions of telomere just by the virtue of tethering telomerase at the telomeric DNA. But you can't let this telomeric extension go on forever. So another major telomeric protein complex, which is called the CST complex, will come in and kick telomerase off the telomeric DNA and terminate that process. But it's also needed to recruit another machinery called a pro-alpha primase to actually complete what we call the C-strand fill-in process. Why you call it C-strand? Because this is the G overhang, and the complementary DNA is called a C-strand, and that's why we need the polymerase alpha primase machinery to do the complementary DNA synthesis. So CST is actually able to do two functions at telomere replication. One is to terminate telomerase activity. Second is to bring this po-alpha primase machinery to do this DNA synthesis. So an important question in the field is really trying to understand how CST mediates these functions, right? So one of the key questions that we want to ask is how actually CST engages single-stranded DNA. As you can see here, both of these processes requires CST to bind to single-stranded telomeric DNA. So the first question is really trying to understand how the CST binds telomeric single-stranded DNA. And our approach is really using a structure function approach, which is really if you get the structure of the CST bound to telomeric DNA, you have an idea how this function comes about. So a little bit of background on CST. So CST is actually made up of three different proteins. The first protein is the CDC1, and then the second protein is TIN1, and the third and last protein is TEN1. They are all of different sizes. And over here is a very simple gel electrophoresis image of a purified protein. Because if you want to study or obtain a structure of the protein of interest, ideally you want to be able to purify and obtain large numbers of your protein of interest in high purity and activity. So here you can see three bands here corresponding to the three subunits of the CST complex. So like I mentioned earlier, we wanted to know the structure of the human CST complex, but people has been trying to solve the human CST complex for decades, and they've only made marginal progress. For example, they are able to solve the smaller subunit TEN1, which is the OB domain, and then they was also able to solve the STIN1 subunit entirely. But really the biggest challenge here is getting a structure of the largest polypeptide CDC1, which they only managed to solve a small segment of it. So the overall architecture of CST as a complex hasn't been known. So to solve the structure of the human CST complex, we resort to this revolution of cryo-electron microscopy. And coupled with single particle analysis, you are able to get high resolution structure of your protein of interest. So you guys might be very familiar with this, but this technique really is using the purified protein, putting it embedded in a special kind of ice called a vitrified ice, which is actually transparent ice, and you can look at it. You can look at these individual samples through the electron beam and capture many images. And then with these images, you want to use the computer to be able to box out individual particles of your protein of interest for downstream processing. So essentially, there are only two major steps in this reconstruction step. So for example, the first step here is really once you box out these individual particles, you want to be able to two-dimensionally align them rotationally and translationally to get what we call the class averages. Because by averaging them, you will reduce the background and enhance the features of your protein of interest. This is presumed that there's only marginal or a single conformation in your protein of interest or in your samples. So as you can see, with two-dimensional or what we call the 2D classification, you're able to sort out the good and bad particles or even junks that you may pick from your cryo-EM imaging. Once you're at this stage, you're able to do what we call the back projection to get the 3D model of your protein of interest. And to do this, you really have to assign the poses of your images to a 3-dimensional model here. So that's how we can use 2D images to get a high resolution structure of your protein of interest. But cryo-EM SPA is really an iterative process, right? As you can see here, you have the purification and analysis of your protein samples. You want to be able to purify your proteins to a high purity and also high activity, and they must be homogeneous. And then you can resort them to negative stain EM, which we're going to talk about later, which is a very quick and easy way to assess the quality of your protein samples before moving on to the more technically challenging and expensive cryo-electron microscope imaging. And of course, if all else works, that would be great. If not, you have to go back to the drawing board, redesign the construct, or figure out how you can best improve the stability of your protein complex before repeating this cycle again. So like I said earlier, even for the human CST, we have to go to, try to look at the quality of our protein samples using negative stain EM. So this is an example of a negative stain of a GRO-EL protein complex. This is not a CST complex. And negative stain, this is how a typical negative stain EM image looks like. So how does it look like for the human CST complex? So subjecting the negative stain image of the human CST to 2D classifications, you can find that we can now see very clear and very consistent features of the CST complex, and you can sort of tell that they all belong to the same kind of conformation, which is good news. And then that really enabled us to move on to try our human CST complex using cryo-electron microscopy. As you can see now that there's a big difference between negative stain EM imaging and cryo-electron microscope imaging. Cryo-electron microscope imaging gives you way lesser contrast, challenging to do image analysis, but actually paybacks in terms of resolution, because you can get very high resolution 3D reconstruction as compared to negative stain EM images. So for the human CST complex under cryo-electron microscope imaging, we found that we are also able to get very nice 2D classification from the cryo-electron microscope data set. And as you can compare to the negative stain EM 2D class averages, they look about the same in size, which is encouraging. But this is not the end, because we still have to get the 3D classification done, which is the natural next step after 2D classification. So what we do in this step here is called a reference-based 3D classification. What we want to do here is really to sort out any heterogeneity in the structure using computation. So you can see here, we can split ab initio model of the CST complex into 4 distinct classes. And from here, you can see 50% of our data set actually allows us to see sort of a blurred vision of the human CST complex. So after further refinement, we get to a 6-angstrom model of the human CST complex. And here is a very typical way to report the resolution of your reconstructed cryo-EM map in cryo-EM single particle analysis. So 6-angstrom is interesting and encouraging at a time, because nobody's seen the human CST complex before. And if we have all the atomic models that has been solved before, one will be able to dock all these available solved structures into a 6-angstrom map and get the atomic model that you need to answer your biological questions. But as you can remember, that the CDC1 subunit of the CST, which is the largest subunit of the CST complex, hasn't been solved before. And at 6-angstrom, we are not able to yet build the atomic structure of the CTC1 protein from scratch. So that really gets us back to this vicious cycle of the research, right? Now the next step is trying to redesign our construct and figure out how we can improve the stability of our human CST complex. So we are really just trying to pull out an old trick from the structural biology field. It's really trying to add in a natural ligand of the human CST. So it turns out, after adding a telomeric single-strand DNA which CST binds, we added a TTA GGG repeated 3 times. We get back, this is a 2D class classification of the human CST with the single-strand DNA added. And you can see most of the 2D class averages are showing the same as the one without the DNA. But what is really unexpected is this huge complex that came out, like, peeking off from the corner of the class averages, which is really interesting. So they really divert our attention to this big guy, trying to figure out what is going on when we added the telomeric DNA. So as it turns out, by adding telomeric single-strand DNA, we are able to trigger a very dramatic assembly of the human CST proteins. As you can see here, this thing is very highly symmetric, but also very interesting. And it really allows us to get a much higher resolution than we got before. We got about 5.2 angstrom from just a very small scale cryo-EM data set. So the next question we want to ask is if we can actually dock, the CST monomer or the individual CST complex that we solved earlier into this big supercomplex, right? So as it turns out, we are able to dock in 10 of that CST monomers. And then that reveals the D5 symmetry of the supercomplex when CST binds to single-stranded telomeric DNA. So D5 symmetry can be really explained by this double crown simplified cartoon over here. You have these, every crown has a 5-point spokes, and they are inverted in this dimeric in a z-axis. We call it inverted dimers. So that really helps us push our resolution down to about 5 angstrom, and really allows us to also now be able to confidently dock in the previously solved structures of 10.1 and STIN1 10.1 into our cryo-EM map. And you can see here, all we need to do is just to use these, what we call the sausages, which is the alpha helixes of the soft structures to guide us into, the structures into the 5 angstrom cryo-EM map. But the resolution of this map is actually limited by the low particle count and the quality of data collected. So after many iterations of troubleshooting and buffer optimization, we finally got to this 3 angstrom cryo-EM map of the CST DNA decamer. As you can now appreciate, there are a lot more features at 3 angstrom as compared to the 5 angstrom map. And one really cool features about the cryo-EM single particle analysis is that we are now able to calculate local resolution, giving us information on which part of the protein complex is rigid and which part is floppy. As you can see here, the rigid or the more intact body of the core body of the CST DNA decamer is blue, which has the highest resolution. And then the legs and the arms, or we call the spokes, are all jogging out. They are more flexible, and that results in a lower resolution reconstruction. So when we reconstruct these to 3 angstrom, we're actually leveraging on, or we're actually imposing symmetry during the 3D refinement step. But there's a danger to it, because when we say we impose symmetry, we are literally saying that every face of the supercomplex looks the same. So every side view of the particle are assigned to every view in a 3D model. So that has a danger of averaging out important conformation differences between the monomers that participate in a supercomplex formation. So what we need to do next is to break the symmetry, right? So the idea is that we are able to now extract individual monomeric CST out and further classify them and find that if we can see any, we can sort them out into different conformations. So this is done through several steps that I'm not going to go into details, but sort of like a image analysis sorcery, where we can now, this is the original image of the decamer CST DNA. And now we are able to pull out individual monomers. As you can see here, these very similar looking the class averages that looks like the one without the DNA included. And with this way, we're able to now use 3D classification to sort out into, let's say, over here, we assume that there are 8 different conformations sitting in this decameric state. And then from here, we can find out that only 50% of this class average allows us to reconstruct the 3 angstrom model here. So you asked, right? Just now it was a 3 angstrom cryo-EM map of the decamer. And now I get back the same, about 3 angstrom. But there's a clear difference between the original map in the decamer and the one that we now get when we in silico purified the monomer from decamer. So as you can see here, even though they report the same resolution, the quality of the map is drastically different. As you can see here, I can't even trace the polypeptide backbone across this map, while in here you can easily trace and find out and build the atomic structure of the polypeptide from scratch. So coming to this point here, we got to this final 3D reconstruction of a real 3 angstrom map of the CST DNA complex. And from this map, you can easily dock in now the even better dock in the soft structures of the STIN1, TEN1, and now even the OB domain that they solved previously from the CDC1. But what's really exciting is that this quality of map allows us to de novo build, which is the build from scratch, almost slightly over 800 amino acid residues of the CDC1 polypeptide. And I think this is probably still one of the largest build from hand so far for a protein complex. So now here is a video of this CST atomic structure that we solved using cryo-electron microscope, including figuring out where now the single-stranded DNA, telomeric DNA is bound. And turns out it's bound to the CDC1 subunit of the CST complex. So as you remember, we used 3 repeats of TTA GGG, but over here, we are only the cryo-EM only revealed 4 nucleotides, which we assigned as TAGG, instead of the full 18 nucleotide of telomeric DNA that we use, right? Well, that could be explained because there's one possibility is that this is the core DNA binding site of the CST complex to single-stranded DNA, and the rest of the telomeric single-stranded DNA is either floppy or in a different register and doesn't average out to give you a very consistent electron density map. So we sort of get a glimpse of where CST binds single-stranded DNA. This is all nice, all good, but how are we sure that, or how can we validate that this is actually where the DNA binds, and if it's the important binding site for the CST complex? So we did a very simple way, is really to look at all the human diseases of CST or CDC1 that abolishes DNA binding. And then now we plot it on a CDC1 atomic structure that we built. And you can see here, these green residues are the mutation residues that we know from human diseases that disrupt DNA binding of CST. And now the model that we get of the single-stranded DNA is very close to the human disease mutations, which is very, very encouraging for structural biologists. But still, we have to validate these, the interactions involved in holding this small piece of single-stranded DNA. So to do this, all we need to do is just to make these CST proteins again, but now introduce mutations that would disrupt, or the mutations that we predict will disrupt the CST binding to single-stranded DNA. And this is a very typical gel shift assay performed by biochemists to figure out the binding affinity of their protein to their DNA of interest. So you can see here, this is a titration of the wild type CST protein. And then this is the substrate that we use, which is exactly the same substrate or the ligand that we use for our cryo-EM map samples. So these are 3 repeats of TTA GGG. This is the unbound state. And if you have a protein bound to it, this thing is going to shift up. As you can see here in the wild type, it retains the original binding affinity of the CST complex. And now we have, and when we introduce these mutants in, you can see now that even though we titrate in a lot of these proteins, the DNA doesn't get shifted up anymore, indicating that they lost their DNA binding ability. And of course, like all experiments, you need to have a controlled mutagenesis. So in this controlled mutagenesis, we introduce mutations that we know are not going to disrupt the DNA binding interactions of CST to single-stranded DNA. So we get the structure of this human CST bound to telomeric single-stranded DNA, but then there's still the big question, there's addressing a big elephant in the room. How did the CST monomer transit to this dichromeric state upon binding to single-stranded telomeric DNA, right? So we have the CST monomer, and how did it form this dichomer upon binding to single-stranded DNA? So we look back at an earlier cryo-EM data set that we know that has a way lower probably efficiency in forming dichomer. And then we did our usual cryo-EM analysis and found there are 2 distinct conformations of the CST monomer here. One is colored in cyan here, and the other one colored in pink here. Very quickly, you will realize that there are actually 2 major differences. One is at the head region, as you can see here, the cyan model has extra density as compared to the pink model. And then now the corresponding arm position that the cyan model doesn't have it, but the pink model has it. So this is what we call the head position, head model or the arm model here, two different conformations, which really leads us to suggest if a C-terminus half of STIN1 is able to do this switch between two sites on the CDC1. But we also know this because we have solved the structure of the human CST complex without single-stranded DNA bound, and also the subsequent one with DNA bound. So we know these two regions are the STIN1, the C-terminus half of the STIN1 subunit. And we also quickly realize that the C-terminus half of STIN1, which is the cyan now in color here, and this is the N-terminus half of STIN1, is actually more loosely bound to the purple colored CDC1 as compared to the N-terminus dark blue colored STIN1 N-terminus half. This really leads us to suggest that this C-terminus half of STIN1 is able to switch between these two positions, the head and the arm. So then obviously the next question is asking if binding to single-stranded DNA will affect the positions of the C-terminus half of STIN1. So if you were to look at the fragment of single-stranded DNA that we solved that was bound to CDC1, you'll find that it's actually very close to the head position of the C-terminus half of STIN1. Presumably that would block the position of the head conformation and forcing the C-terminus half of STIN1 to bind only at the arm position. So with this, it led us to this assembly model of the CST DNA dichomer formation over here. So without DNA, the STIN1 C-terminus half is actually bound to this head position and is jogging out here, preventing the inverted dimerization, as you can see next, where if you plus the DNA now, it will go to the arm position, making way for the inverted dimerization to occur. And once this dimerization occurs, it will then let the tetramerization proceed. Eventually, if you make enough of this tetramerization, you get this closed-end oligomerization forming the dichomeric supercomplex structure. And of course, this is initially all a model, a hypothesis, and we have to go set out to get the intermediate states or even catching the intermediate states in action. So with negative stain EM, we're able to catch most of these intermediate states in action. As you can see here, this is the CST monomer, and you can even see the little head jogging out here, which is the C-terminus half of STIN1. And then of course, plus the DNA, we didn't manage to capture it. Presumably it moves it oligomerized so fast to the dimerization and then the tetramerization state before it gets to the dichomer state. So an interesting observation is that we notice that the two small fragments of single-stranded DNA in the inverted dimer is very close to each other. As you can see in the video here, that these are the gold or yellow colored 4 nucleotides bound to the CDC1 subunits of the respective monomers in the dimer form, right? They're so close to each other that we are thinking that, the DNA that we use, which is actually a single 18 nucleotide of TTA GGG, is actually able to staple the two inverted dimers together. So it leads us to refine our model a little bit, saying that the single-stranded DNA is actually important for the dimerization process. So what is next for the CST dichomer story? So in our lab now, we are really trying to understand the structure functional studies of the human CST bound to the pole-alpha primase machinery that I mentioned earlier, right? Where the CST, like I said earlier, was involved in two major functions in telomere replication. One is to terminate the telomerase activity. The second one is to recruit this pole-alpha primase machinery to form the C-strand, to mediate the C-strand fusing process. So it's of our interest to understand how this pole-alpha primase interacts with the CST monomer or even a decamer to form the co-complex that's important for the C-strand fill-in. And this project is spearheaded by one of our graduate students, Qi Xiang, in this project. And to do this, Qi Xiang actually have to first solve the human pro-alpha primase structure first, because this hasn't been solved before, and using cryo-electron microscope, which the same steps that we mentioned for the human CST complex, he was now able to capture the pro-alpha primase machinery doing the RNA DNA transfer, and he caught it in action here. You can see we now got to about 3.5 angstrom model, and we are able to fit in most of the previously solved structure now, and we are one step closer to understanding how the pro-alpha primase is able to transfer the RNA primer to DNA synthesis. So with this, I would like to end my talk here. I'd like to acknowledge Tom, which I did my postdoctoral work with. He successfully transformed me to a biochemist and a structural biologist from an experimental single molecule biophysicist. And now in UW-Madison, I'm running my own lab that uses both single molecule biophysics and structural biology to understand questions in human telomere biology. So with this, I'll be happy to take any questions. Thank you.
Okay, thank you, CJ. Great talk. So on to have a question. Can David? Can everybody talk, or you have to give?
No, go ahead, Antoine. Maybe you.
Yeah. So I mean, anyone who has
just want to Antoine. Just want to say that if you are shy to ask directly your question, you can use the chat box as well.
Okay, right. Yeah. So please feel free to ask a question, and as David mentioned, you can also type your question on the chat box. Maybe while waiting, let me ask a question. So you have several conformation of your subunit, right? So you break the symmetry. So how many supposed to be conformation out of that? I mean, you talk about purification conformation process, but it's not very clear how you did it.
Well, maybe I'll go back to the slide. So Antoine, are you referring to this?
Yes. So you have a structure which fully imposed symmetric. All the 10 subunits. So now, how do you break the symmetry?
So when we pose a D5 symmetry, right? So we already align the particles to a reference D5 symmetry,
which is average, eh? Yeah.
Right. And then but then at the D5 symmetry, we have to define. You have to use one monomer as a reference point. So we have to do actually in here. We have to symmetry expand based on D5. So what the computer does is actually they will rotate the coordinates 5 times, and then another 5 times in an inverted manner. So then very quickly you expand your data set by 10-fold because it's D5, right?
I see.
And then once you have that, you have to do what we call this image subtraction, just to clear the background a little bit. So that's why you can see here in the lower panel here that this is the one after we do what we call the particle subtraction. That is actually the same thing, but now we retain. Now you can see we only be able to see one monomer out of the 10 monomers in this decameric structure. So,
right, but now you assume that they're not symmetric anymore. Then how many conformations do you think they're likely to be?
Well, the how many conformation question actually comes to here. So it's not how many conformations, but rather how many of these proteins are actually intact, or allows us to solve to high resolution as compared to. So you can see here that we split it to 8 conformations, right? Or we say, I wouldn't say conformation, I'll say 8 classes in an image classification. And then you can see here one image stood out because that actually gives the highest resolution, while you can see the rest here, like one class here and this class here, they all give very poor features. So they could be either very flexible, or they don't give enough particles to reconstruct to high resolution, or they could be damaged but still participate in the oligomerization process.
But it could be that okay. So here you basically you have a population of different conformation, and one kind of stand out at 50%, 49%. But it could be that coexistence of different conformations. I mean, all the structure, all the subunit cannot be in the same conformation at the same time. You know, I mean, out of the 5, if one take one and the other have to be other, and then maybe dynamically that change over time. So
I think that could be it. But if there's another conformation that is that allows us to solve using cryo-EM, I think it would have shown up here. It's like I say, it could be either we don't have enough particles, but I don't think that will be the case. I think more likely is that unless it's way off out of the mask, that it doesn't sit inside the mask and solve it properly. But I think at this point now, what we are thinking is that this is either poorly resolved due to very flexible monomer. But then also going back to your question, I think that the way to do this is we need to know how these good, or what we call the good monomers are sitting relative to each other, which we don't have the answer yet.
Okay, great. I think Dahai has a question, I guess.
Hi, CJ. Very nice to see you.
Hi.
Thank you for
Nice
coming
to
back
see you in
again.
person.
Give a more detailed talk. I remember during the AP candidate talk, it was very brief, and we don't have a chance to ask questions.
So
yeah. 2 small questions. One is the AFM experiment. I remember in the early slides you had a few AFM images of the DNA complex with what is the name of the molecule? Those were dry images, or you have them in solution?
These are dry images. These are not in solution AFM imaging.
Right. I'm just curious. I know there's technique advanced that you can do AFM imaging in solution or even real time, sort of.
Yes, that's true. You can actually now do movies of AFM imaging in real time. But that's actually if you look back at the literature, there's only one group that consistently being able to do that, and that was actually by their home built AFM.
Right.
So I think it's still the technique is still not yet mainstream.
Interesting. Okay, cool.
But yes, that is a very very cool technique that actually exists for almost more than 10 years now.
Right. So second question, have you ever tried the CryoSpark 3D flexible refinement? That they haven't officially launched, but I'm just curious, have you tried it with your Dagmar CTS? Would you see interesting motions, local motions that, I don't know, suggest any functional significance?
Right. So we did it for the CST Dagmar. I think you're referring to the 3D variation analysis, right?
Yep.
Right. So we did it on a CST Dagmar, and I think the only place I published it was on Twitter. But it seems like we can see, using 3D VA, we can see that the spokes or the legs of these CST that actually breathing in inverted pattern. So that means, for example, if I can see, if I can capture this guy, maybe there's another slide that might be easier for me to explain. This one. So you can see these are the 5 legs, right, from one side. Yeah. Right. So what we saw from 3D VA is that if one leg go out, the other leg will come in. So although this is very interesting, we don't have a clear path to try to understand, put it in the context of a function, but clearly it has a role to play in the flexibility of the legs.
Right, because they are not really in contact with each other.
Right, but the body itself is pretty rigid, so we don't see any. At least the PCA analysis doesn't show up any movement in the body of the CST Dagmar, but the legs and the spokes are actually moving quite a bit. So it's actually a very malleable super complex. It's like breathing.
Cool. Yeah. Cool. Antoine, can I ask one more question?
Yeah, please.
Yeah, so in the dimer form, you capture the DNA, which is only 4 base, and the rest are sort of disordered, I assume. Yeah, in the dimer form, the 2 DNA are inverted, facing each other, and oriented each other, sort of.
Well, we try to draw orientation, so it's along this, so it's inverted in a sense because they're
Are you
inverted
sure
by-
they are in the same? They are sharing the same strand, or they could be from different DNA?
So we did actually MSAs. So one way we can figure this out is that we can do gel shift assays, correct? So we can scramble this register or the middle register or the last register and see how it hurts the DNA binding affinity. So and that was actually published in our paper with this cryo-EM model that this is actually the CST is likely using a single piece or the same piece of DNA to breach the dimers.
Right. But
So-
between the dimers, they must be different DNA strands, I assume.
Well, as of what we saw, we know it's TAGG. And then since the TAGG only happens once per repeat that we used, so we think that this is going to be the first and a third repeat from, for example, the pink monomer here is binding to the first repeat and the blue monomer here is binding to the last repeat. And then when we scramble the sequence over in the second repeat, we see no consequences on the DNA binding affinity of CST. While the first and the third repeat actually has a lot of impact on how the CST binds the DNA.
Yeah, I mean, I really know very little about these telomere sequences. At some point, this DNA has to be double-stranded, are they?
Right, but the CST don't bind double-stranded telomeric DNA. Yes,
Yeah.
but a majority of the telomeres are actually double-stranded DNA, and like a couple of hundreds of nucleotides, it ends with a 3' overhang. And this is actually where most of the molecular actions are taking place.
Right, interesting. Okay, thank you.
Thank you.
Just to follow up the high question. So in this model, right? So you see from the bottom, it goes from the right to the left. And then from the from TTAGG, from the top, it goes from the left to the right. So is the hand, two hand, right, of the CST, is the right hand, oh, sorry. It's the one that on the right of the top is the same as the one that on the left. The same as the right of the left or not? I don't know the symmetry of that. So you see what I mean? Because DTA, GGG run from the left to the right for the top one, and then right to the left from the bottom one.
Right.
So what's so it is the same unit, or because you draw the two hand more or less the same? So
right. The cartoon is there's only so much we can.
Okay. So it's the same direction then, right?
It is the same direction because the monomer is derived from the decamer.
Yes. So what you're saying that the middle repeat is not important for the recruitment.
Well, it's not important for the DNA binding.
So so can you have the recruitment with 2 repeats?
No, it'll be too short because if we delete the middle repeat, it'll be too short to breach. So we have shown that also in our publication.
But so that means you can put anything about 5, 6 nucleotide in between, then that's enough to breach, right?
That's correct, and that's what we did, and show that it doesn't hurt the affinity of CST to telomeric DNA.
Okay. So, but then I saw the complex, they have a very big hole in the middle. What is this used for?
Well, I don't know. We are trying to find it out. But I think one interesting model that I did not show here is that if CST can form a decamer at a single telomeric overhang, then it has the potential to recruit multiple pro-alpha primates per overhang. And if you remember that the C-strand fill-in process, which is to make that single-stranded DNA into double-stranded form, right, is actually like a Okazaki fragment. It's basically a lagging strand synthesis. So if CST is able to form a decamer and recruit multiple pro-alpha primates to a single-stranded DNA overhang, it would rapidly fill up that C-strand fill-in process. So this is one of the major research direction our lab is undertaking now to figure out how pro-alpha primates would occupy a decameric CST and how it affects the pro-alpha primates enzymatic actions.
Okay. So I guess if you have a long single strand, it would wrap around, right?
Right. Yeah. Well, we get that question a lot, and then we also tried really hard to reconstitute CST decamer on a very long piece of telomeric DNA, but it turns out it's actually thermodynamically a poor solution because, as you know, telomeric DNA is repetitive. Trying to force it to sit in a very orderly manner is not an easy process. Anyone that worked with nucleosome already will find very painful to work with, much less a hexamer repeating sequence like that. So yes, we see if we put a long piece of telomeric DNA with CST, we see a lot of protein aggregation. So there may be a chaperone that's involved in assembling CST at the telomeric DNA, or maybe it's a very carefully titrated process.
I see. Okay, so any other question? Yes, please.
Hi, thanks for the great talk. So just one small question with regards to the assembly model for the CST DNA decamer formation. Just roughly, like, do you know how fast these processes occur, like the dimerization and so on? How fast do these occur?
Oh, that's a great question, but I only can answer you based on the negative stain images that we got. As you can see here, we actually did a population analysis on these intermediate states, and it turns out that we capture a lot of the tetramer here. You can see this is 4 copies, and then there's 2 copies. We don't see it with the DNA bound, so that is a very fast step. I think the second fast step is actually from the tetramer to this decameric closed-end structure. But saying that, negative stain EM is actually a dry imaging process. You need to dry your samples, and that's actually a very harsh condition for such a fluffy or structured complex like this. So you can imagine that if you can form, for example, a tetramer and then become an octamer, the octamer might not be stable enough to remain intact upon drying. So it might break apart, but at least it break apart into 2 tetramers. So, but we do think that the fast or the speedy step is actually when it binds to single-stranded DNA, it should rapidly move to dimer form, because that would actually stabilize the position of the C-terminus half of STIN1, right? Because if that is not fast, it can very easily revert back to the no DNA conformation. So, but that's a good question, and that's also if we know which are the rate limiting steps, that could be very useful in trying to control these individual steps of oligomerization.
Thank you.
Any other question, please?
Hi. It's a question with regards to the Josh's project, where the shelterin binds to the 600 base pair telomeric DNA. I missed it. So is the shelterin mostly binding to the ends of the DNA, or is it binding everywhere, the complex?
You mean this the
Yes.
shelterin complex here?
Yes.
So what we've seen so far is that if we were to introduce in shelterin core complex is actually just I think the cartoon here is TRF2, TN2, TPP1, POT1. So it's just 4 subunits. But this complex has both the single-stranded and double-stranded DNA binding capability. And what we are seeing so far is that this complex is only localizing at the end of the telomeric DNA construct that we gave.
Okay. So it's close to the single-stranded region.
Well, we don't have, AFM doesn't have the resolution to resolve a single-strand DNA of this size. It can resolve single-strand DNA, double-strand DNA in AFM just based on the rigidity of the polymer. But this is so small overhang that we can't see it actually. So, but then we can see it just by, for example, POT1 is a protein it binds to. You can see that brighter spot at the end of the DNA. But at least we don't see 2 bright spots at 2 ends, both ends of the
Makes
single.
sense. Makes sense. Okay.
Right.
Thank you.
Thank you.
Yes, more questions? What? Okay, if not, then let's thank CJ. His amazing talk, and good luck for setting up the lab.
Thank you.
It's a great pleasure to have you. Maybe come back again. Yeah.
Hopefully, I'll do the next one in person.
Yes,
Okay,
yes,
good.
yes.
Right.
That'll be good.
More
This will be
data
great.
from my lab. Okay. Okay. Take care,
Thank
everyone.
you very much,
Thank you.
CJ. Have a nice evening because it's late in US now. Thank you for coming and joining us that late.
Thank you.
Take care.
Thank
Thank
you.
you.
Thank you,
Everyone.
everyone. Have a good day.
Bye bye.